Hello, this is Jonathan Roberts, and the purpose of this video will be to discuss the differences and similarities between cellular respiration and photosynthesis. In this video, you will see a side-by-side comparison of the two biological processes. The explanation will begin with a general overview of the two processes, and then focus in on the more specific stages. During the conclusion, I will sum up all that I have talked about in a simple T-chart. With that being said, let us begin. As you can see, these are the two equations of each of the individual processes. Photosynthesis reads carbon dioxide plus water plus energy equals glucose and oxygen. Cellular respiration reads glucose plus oxygen equals carbon dioxide, water, and energy. These equations complement each other, as photosynthesis uses the products from cellular respiration as reactants, and vice versa. Another difference that can be spotted right away is the organelle at which each of these procedures takes place. Photosynthesis takes place in the chloroplast, as you see on the left, which is an exclusive organelle to plant cells. Cellular respiration does not take place in the chloroplast. Instead, it takes place in the mitochondria, as you see on the right. Both organelles resemble the shape of a bean, and both organelles have an inner and outer membrane. Cellular respiration occurs in animals and humans. This is where you'll find the mitochondria. Photosynthesis occurs in plants and certain bacteria. This is where you will find the chloroplast. In photosynthesis, there are only two stages, the light-dependent reactions, which happens first, and the light-independent reactions, the Calvin cycle, which is the second and final set of reactions in photosynthesis. There is double the amount of stages in cellular respiration. The four stages of cellular respiration in order include glycolysis, pyruvate oxidation, the citric acid cycle, and the electron transport chain. A key similarity that we can see when analyzing these different stages is where ATP is formed. In photosynthesis, ATP is only formed in the light-dependent reactions. During cellular respiration, however, ATP is formed in glycolysis, the citric acid cycle, and the electron transport chain. The difference, however, is how this ATP will be used. In photosynthesis, the ATPs are stored rather than used for other purposes, like in cellular respiration. From this, and the equations of each of the processes, we can assign a delta G value to each of these procedures. Photosynthesis has a positive delta G, meaning it is an endergonic reaction. Cellular respiration, on the other hand, has a negative delta G value, meaning it is an exergonic reaction. This can be further portrayed by the process's production and consumption of glucose. In photosynthesis, glucose is created, meaning bonds are being formed, resulting in stored energy. In cellular respiration, bonds are broken, which means that glucose is being broken up, and this results in a release of energy. All of these points emphasize the fact that both of these biological processes coincide with each other. The both of them keep each other in motion. Now it's time for a more in-depth analysis. Contrary to first glance, the light-dependent reactions of photosynthesis and the electron transport chain of cellular respiration are actually quite similar. To begin, they both have an ATP synthase located on the right of each of the diagrams displayed on the screen. In the light-dependent reaction, we can see that the ATP synthase is right here, and in the electron transport chain, we can see that it is right here. They're both relatively in the same spot. Both of these stages have an ATP synthase because, as you may have guessed, ATP is created. The ATP synthase operates by making use of the high protein concentration gradient. In the light-dependent reactions, you can see that the majority of the hydrogen atoms are inside the thylakoid lumen, and as a result go through the ATP synthase into the chloroplast stroma. In the electron transport chain, however, there is a large amount of hydrogen atoms in the intermembrane space that naturally move from the ATP synthase into the matrix. In the thylakoid lumen, there are a lot of protons, and they go through the ATP synthase into the chloroplast stroma. In the electron transport chain, they're in the intermembrane space right here, and they go into the matrix of the mitochondria. Now, in terms of electron flow, in both scenarios the electrons move from left to right across the membrane for a certain purpose. In light-dependent reactions, the electrons move to eventually power the NADP plus reductase. In the electron transport chain, however, electrons are moved across the membrane in order to reduce oxygen and create water. In the light-dependent reaction, we see that water is being stripped of its electron, and it moves across the membrane all the way over to the NADP plus reductase. In the electron transport chain, electrons move through the membrane in order to get to oxygen and reduce it to water. As I just mentioned, water is a key player in both of the processes. However, in photosynthesis, it is broken down by the water-splitting enzyme. The opposite occurs in cellular respiration. Water is created at the end of the electron transport chain. The electrons also come from different sources depending on the process. Electrons are contributed from the splitting of the water in the light-dependent reaction, while in cellular respiration, they are obtained from the NADH and the FADH2. The last of the significant differences is derived from the name of these two stages. Photon is a necessary key player in the light-dependent reactions of photosynthesis, hence the name. Cellular respiration can occur regardless of whether or not light is present. This basically sums up a basic comparison of the two stages. Depending on the process, the electrons come from different sources. In the light-dependent reactions, we can see that the water-splitting enzyme is splitting the water to take its electron through the membrane, while on the other hand, we have the electron transport chain, which takes NADH and turns it into NAD, splitting it of its electron and going through the membrane from there. And also, it uses FADH2, excuse my poor writing, as a electron contributor as well, which is different from the light-dependent reactions. And another difference is that the light-dependent reactions, they need light to function, and the electron transport chain doesn't. The next pair of stages in photosynthesis and cellular respiration that are similar are the light-independent reactions and the citric acid cycle. This is going to be a little more brief than the last slide. The first similarity we can spot is both of their ability to function in a cycle-like formation. Also, both begin with a certain molecule and throughout the cycle need to reproduce that certain molecule in order to ensure that the cycle will continue to operate. Oxaloacetate is the beginning molecule of the citric acid cycle, and it needs to be replenished in order for the cycle to continue itself. It's the beginning step of the citric acid cycle, and it's paired up with the acetyl CoA to make citrate and to continue the rest of the cycle. In the light-independent reaction, we can identify that the beginning molecule is RuBP, and Ribisco, the enzyme, pairs it with carbon dioxide to create 3-phosphoglycerate, and from there it continues the rest of the cycle. The main difference that we can note is regarding the use of carbon dioxide. In the light-independent reactions, carbon dioxide is attached to the RuBP right here, and they attach itself to form the 3-phosphoglycerate via Ribisco, the enzyme. While in the citric acid cycle, carbon dioxide is released as a byproduct, and we can see that right here in the very beginning of the cycle. This shows how the two processes complement each other. One uses carbon dioxide while the other produces it. And now to wrap all of this up, to summarize all that I've explained, I've completed a chart that we were given in class, which compares and contrasts the two biological processes. There are many key players in both of these procedures, so I'll explain their functions in both. In photosynthesis, water is split via the water-splitting enzyme, and it is a reactant. In cellular respiration, water is created at the end of the electron transport chain, and it is a product. Oxygen is another key player. It is released into the environment as a byproduct by photosynthesis, which provides humans and other animals with the oxygen they need as a reactant during cellular respiration. In cellular respiration, carbon dioxide is released as a byproduct, and in photosynthesis, it is used as a reactant. NAD+ is also another key player, which is used in both biological processes. In the case of photosynthesis, NAD+ is used in the light-independent reactions to reduce 3-PGAs and form G3Ps. NAD+ in cellular respiration is used to create NADH, which in turn creates ATP. Electrons power the NADP+ reductase in the light-dependent reactions, while in the electron transport chain, they are used to reduce oxygen and create water. Protons are very important in both processes as well, as there is a high proton gradient which powers an ATP synthase that is present in both the procedures. The ATP generated in both of these procedures are used in different ways. In cellular respiration, ATP serves as an energy source that is used to power other functions of the body, while ATP is conserved in photosynthesis to power the light-independent reactions. These light-independent reactions, the Calvin cycle, uses ATP to produce glucose, one of the products of photosynthesis. Meanwhile, in cellular respiration, glucose is consumed in glycolysis in order for the entirety of cellular respiration to run its course. Glucose is a reactant in cellular respiration. Both of the two processes take place in and near two specific organelles. The chloroplast is the site where photosynthesis takes place, while the mitochondria is where cellular respiration occurs. There is a flow of electrons in both events. In photosynthesis, it takes place on the thylakoid membrane, and in cellular respiration, it takes place on the cristae of the mitochondria. And finally, the high proton concentration is in the lumen of the chloroplast and the intermembrane space of the mitochondria. This concludes the video. Photosynthesis and cellular respiration are both necessary to life on Earth, and each of them could not function without each other. That is why they are so similar, and at the same time, as explained in the video, very much different. It is this harmonious interaction between the two of them that makes them so efficient at what they do. This video's purpose was to be informative and educational, without being too long and tedious, and I hope it proved to do just that. Thank you for watching.